expression can be regulated at the amount of protein and at the level of protein activity. But transcription is the primary side of regulation. This lecture is divided into two parts. In the first part, I will introduce you regulation of prokaryotic gene expression. In the second part, I will describe to you regulation of eukaryotic gene expression. In prokaryotes, the major site of regulation is transcription. There are two typical types of transcriptional control. One is regulation by activator and repressor proteins, as shown in regulation of operon and operon. An operon is a cluster of coordinately regulated genes. Second one is attenuation control, as shown in histidine operon and operon. Here is a gene, and this is a promoter operator region, and this is RNA polymerase. Without any repressor present, RNA polymerase can initiate transcription. But when a repressor protein binds the operator, RNA polymerase cannot initiate transcription. Gene expression is turned off. On the other hand, positive control requires an activator. Here is the gene, and here is promoter. Without an activator, RNA polymerase cannot initiate transcription. Gene expression is off. But when an activator protein binds a specific site on DNA, RNA polymerase can initiate transcription. Gene expression is turned on. The E. coli operon use both repressor and activator. When glucose is not available while lactose is available, they still can metabolize lactose. For this, they require extra proteins to metabolize lactose. For example, they need permease to import lactose and beta-galactosidase to cleave lactose into two monosaccharides, galactose and glucose. Here is a operon. This is a promoter and this is the operator. They are small DNA segments. Lac-I encodes a repressor protein. Lac-Z encodes beta-galactosidase. Lac-Y encodes permease. Lac-A encodes the third enzyme whose function is not clear. Expression of operon is controlled by two proteins. One is repressor that binds operator, and here is operator. Second, cap protein that binds capsid, upstream of promoter. Here is capsid. Here, two sugars, lactose and glucose, provide two signals, and they work through these two proteins to control gene expression. Specifically, lactose induces gene expression by binding the repressor and preventing the protein from binding to the operator. Glucose represses gene expression by lowering the level of cyclic AMP, which prevents cap protein from binding capsid. When both lactose and glucose are present, both proteins do not bind DNA. As in this case, there's a minimum gene expression. Only when lactose is present and glucose is absent, high level of gene expression will occur, as shown in this case. When lactose is present and glucose is absent. When glucose is absent, the cyclic KMP level rises. Cyclic KMP binds and activates cap protein. Cap protein then binds capsid and promotes RNA polymerase to initiate transcription. Now, high level of LAC gene expression are achieved. When glucose is absent, the cyclic KMP level rises. Cyclic KMP binds and activates cap protein. Cap protein then binds capsid and promotes RNA polymerase to initiate transcription. Now, high level of LAC gene expression are achieved. Histidine operon encodes enzymes that synthesize histidine. If histidine is available, the cell does not need to synthesize histidine. The histidine operon is inactive. When histidine is not available, the cell has to synthesize it by itself, and genes of histidine operon are turned on. The histidine operon is regulated by attenuation. Attenuation leads to premature termination of transcription. It depends on the fact that translation and transcription are coupled in bacteria. Here is histidine operon. The 5' regulatory region of the histidine operon encodes a 16 amino acid leader peptide, including 7 consecutive histidine codons, followed by a transcription terminator. When histidine levels are high in the medium, ribosom proceeds through the leader sequence to the stop codon. This results in formation of EF stem loop. It is attenuator, which serves as a termination signal to terminate transcription of structured genes following the stem loop. When histidine levels are low, the ribosome moves through the histidine codon more slowly. Installed ribosome allows for the formation of an alternative RNA secondary structure that prevents formation of EF stem loop. Therefore, RNA polymerase can continue transcription into the histidine operon structured genes. CHIP operon is controlled by both repression and attenuation. Here is CHIP operon. This is operator, and this is promoter. CHIP A to E encodes structured proteins that are needed to synthesize tryptophan. CHIP R encodes repressor. CHIP L encodes leader RNA transcript. The transcripts includes 4 short sequences, each of which is partially complementary to the next one. The leader RNA transcript provides a stem loop for attenuation. When tryptophan levels are high, tryptophan binds repressor, which now can bind operator and block transcription of CHIP-L and structured genes. This animation summarizes the mechanism by which repressor control expression of CHIP operon. When tryptophan accumulates to excess, it binds repressor. The repressor now can bind the operator and block transcription of CHIP-L and structured genes. This slide illustrates how attenuation controls expression of the CHIP operon. When tryptophan levels are high, the ribosome position allowed the formation of 3-4 stem loop, which serve as a role-independent terminator. The terminator terminates transcription in RNA polymerase detached. When tryptophan levels are low, ribosome stores at the tryptophan colon and prevents formation of 3-4 stem loop. Therefore, no terminator sequence formed. RNA polymerase continue transcription through structured genes. Now let's move to part 2. The first step in gene expression, transcription, is the primary site of regulation in both prokaryotes and eukaryotes. In eukaryotes, gene expression also involves extensive post-transcriptional processes, as well as actions that influence access to particular regions of the DNA. In part 2, we will focus on 3 topics: trans-acting and cis-acting elements, control of access to DNA, and post-transcriptional control. Regulatory sequences of DNA are usually embedded in the non-coding regions of the genome. These sequences are called cis-acting elements because they influence expression of genes only on the same chromosome. A trans-acting element is the regulatory molecule itself, which can diffuse through the cell from the site of synthesis to its DNA binding site. A transcription factor is a trans-acting element. The major cis-acting elements are promoter and enhancer. Upstream promoter region includes these 3 sites. First is CCAAT box at position around -75 that binds transcription factor NF1. A GC-rich sequence that binds SP1, and TATA box at position around -25 that bind TF2D. These 3 are general transcription factors. Enhancers are sites on DNA that bind specific transcription factors. Enhancers have several characteristics. They may be up to 1,000 base pairs away from the gene. They could be located upstream or downstream or even within the intron. For example, the immunoglobulin heavy chain locus has an enhancer located in a large intron. Third, the orientation of the enhancer is not important. Fourth, enhancers can appear to act in a tissue-specific manner if the corresponding specific transcription factors are present only in certain tissues. Fifth, enhancers may be brought close to the promoter region in space by bending of the DNA molecule. Here is DNA, and here is the enhancer, and this is a specific transcription factor. By binding enhancer, specific transcription factor can bend DNA molecule and bring the enhancer close to the promoter. Given a wild-type enhancer sequence and a series of deletions along with the corresponding reported activities, students are required to map out the enhancer region on DNA. This is called enhancer mapping. For a specific example, please see practice questions. A trans-acting element is the regulatory molecule itself, which can diffuse through the cell from its site of synthesis to its DNA binding site. Transcription factor is a trans-acting element. In molecular biology, a transcription factor is a protein that controls the rate of gene transcription, by binding to a specific DNA sequence. A defining feature of transcription factors is that they contain at least one DNA binding domain. Other proteins, such as coactivators, chromatin remodelers, histone acetyltransferases, histone deacetylases, kinases, and others, while also essential for gene regulation, lack DNA binding domains, and therefore are not transcription factors. Two types of transcription factors can be distinguished: general transcription factors and specific transcription factors. General transcription factors must bind to the promoter to allow RNA polymerase II to bind and form the initiation complex at the start site of transcription. TF2D, SP1, and NF1 are general transcription factors. Specific transcription factors bind to enhancer regions and modulate formation of initiation complex, thus regulating the rate of initiation of transcription. Steroid receptors and CREB protein are specific transcription factors. Transcription factors typically have two domains. One is DNA binding domain, which recognizes a specific DNA sequence of the promoter or enhancer and binds to it. Second, activation domain, which interacts with other proteins and thereby increases the rate of transcription. The activation domain allows transcription factor to bind to other transcription factors or interact with RNA polymerase II to stabilize the formation of the initiation complex, or recruit chromatin modifying proteins such as histone acetylases or deacetylases. DNA binding domains contain common structure motifs. For example, zinc finger is a motif found in steroid hormone receptors and PPARs. Leucine zipper is found in CREB. Helix-turn helix is found in homeodomain proteins. Here is clinical correlation. Klein-Waardenburg syndrome is caused by mutation in PEG3, a homeobox gene. Symptoms include dystopia cantharum. It refers to lateral displacements of inner corner of the eye. Lateral displacements of inner corner of the eye. Pigmentary abnormalities, limb abnormalities, and congenital deafness. In eukaryotes, general transcription factors such as TF2D, SP1, and NF1 must bind to upstream promoter regions to allow RNA polymerase II to bind to promoter and form the initiation complex at the start of transcription. Here is an example of general transcription factor sites around the promoter region. TF2D binds Tata box first, which recruit TF2B, followed by RNA polymerase II, TF2E, and TF2H. In some cases, this is sufficient to form the initiation complex, and the RNA polymerase II can start basal minimal transcription. In some other cases, NF1, CBP, and SP1 are required to form the initiation complex. Specific transcription factors work along or with other proteins in the complex by promoting or blocking the recruitment of RNA polymerase to specific genes. Activator proteins promote transcription. Repressor proteins block transcription. DNA sequence to which the specific transcription factor binds are called response elements. Most of the response elements are located outside the promoter region, clustered to form an enhancer region that allow control of gene expression by multiple signals. Hormones provide such a signal. Examples include glucagon and cortisol. Glucagon controls gene expression by phosphorylation of CRAB, a transcription factor. Cortisol controls gene expression by binding a steroid hormone receptor. Here is an example of how steroid hormone controls gene expression. Steroid hormone receptor is a nuclear receptor. In this case, it is found in the cytoplasm. When steroid hormone arrives, it binds the receptor. The activator receptor enters the nucleus, bind the enhancer region of the gene, and increase the transcription of the gene. Increased mRNA leads to increased protein synthesis. PPARs are nuclear receptors. They are specific transcription factors, members of the zinc finger protein family. Their natural ligands are fatty acids. Thiazolidinediones are anti-diabetic drugs. They bind a PPAR gamma. They increase storage of fatty acids in adipocytes, decrease the amount of fatty acids in circulation, and therefore increase glucose consumption. Clofibrate is a drug for hyperlipidemia. It binds PPAR alpha. It increases beta oxidation, increases HDL content, and increases lipoprotein lipase activity, and decreases triacylglycerol secretion. The second way to alter gene expression is to control access to DNA. There are two ways to control access to DNA. First, chromatin remodeling. Chromatin remodeling can be achieved by two major complexes. One is histone-modifying complexes. Second one is ATP-dependent chromatin remodeling complexes. For NBME, we will focus on histone-modifying complexes. They modify histone by acetylation, methylation, phosphorylation, and ubiquitination. Acetylation occurs when an acyl group is transferred to a lysine residue. Methylation occurs when methyl groups are transferred to a lysine residue. Acetylation favors gene expression. Methylation of lysine residues at position 4 and 36 of histone 3 also favors gene expression. In contrast, removal of acyl group by HDAC favors inactive chromatin and decreases gene expression. Methylation on lysine residues at position 9 and 27 of histone 3 has a similar effect. Second way to control access to DNA is DNA methylation. A methyl group is transferred to a cytosine base. This occurs in CG-rich regions. This reaction is catalyzed by methyltransferase using SAM as a methyl donor. As you may recall from lecture B01, SAM is derived from methionine. Evidence supports DNA hypermethylation silences gene expression. In eukaryotes, RNA molecules are processed before they leave the nucleus. Most eukaryotic genes contain introns, which are spliced through RNA splicing. Alternative splicing generates different forms of proteins from the same gene. For example, fibronectin mRNA made in fibroblast is longer than that made from the same gene in hepatocytes. Some cells can alter the RNA sequence through RNA editing. RNA editing requires a guide RNA. Guide RNA has three features. First, it is complementary to the target RNA. Second, it carries sequence to be inserted. Third, it carries a poly-U tail. For example, this is RNA to be edited, and this is the guide RNA. Guide RNA carries 2 extra A. After editing, the RNA carries 2 extra U inserted between C and G. In human enterocytes, ApoB gene carries CAA in the middle of the original RNA sequence. Enterocytes produce a specific cytidine deaminase that removes an amino group from cytosine base within CAA. As a result, CAA is changed to UAA, a stop codon. In enterocytes, ApoB genes yields a shorter protein called ApoB48. B48 is shorter than full-length ApoB100 produced in the liver. This is another form of RNA editing. RNA transport can be a rate-limiting step in gene expression. For example, HIV is a retrovirus. Once inserted into host genome, the viral DNA is transcribed as one long RNA molecule by the human cell RNA polymerase II. The transcript is then spliced in many different ways to produce over 30 different species of mRNA, which in turn are translated into a variety of different proteins. In order to replicate viral genome, entire, unspliced viral transcript must be exported from nucleus to the cytoplasm, where they are packaged into viral capsids. How does the virus export full-length RNA? This is a challenge. The key is this REV gene. The REV gene encodes a Ref protein. This protein binds a specific sequence called REV responsive element, RRE, located within a viral intron. The Ref protein interacts with a nuclear export receptor, which directs the movement of viral RNA through the nuclear pore into the cytoplasm. This is a Ref protein. It comes into the nucleus, bind the RRE, and bring the full-length viral transcript into the cytoplasm. The fifth method to control gene expression is to control stability of mRNA. In eukaryotic cells, poly-A shortening occurs slowly, but once it reaches a certain degree, 2 degradation events occur. First, 5' decapping and 5' to 3' degradation occurs. Second, 3' to 5' degradation occurs by exonuclease. Degradation of mRNA can be facilitated by RNA interference or RNAi. It is mediated by short non-coding RNAs called microRNAs. A microRNA is initially synthesized as primary microRNA transcript, as shown here. After RNA processing, it is shortened, giving rise to pre-microRNA. Then it is exported into the cytoplasm, where it is cleaved by an enzyme, dicer. A mature strand is loaded onto a protein complex called RNA-induced silencing complex, or RISC. The mature strand hybridizes with a complementary sequence on a full-length target RNA, bringing RISC to the mRNA, which blocks translation and promotes poly-A tail shortening. Therefore, microRNA blocks translation and decreases mRNA stability. For translational control, I'm going to show you two examples. One example is to use the stem loop in control of ferritin translation. Ferritin is a protein that stores iron when iron levels are high inside cells. Here is a ferritin mRNA with a stem loop at the 5' end. When iron levels are low, a protein called IRP binds the stem loop at the 5' end and blocks translation. When iron levels are high, iron binds IRP and will release the protein from the stem loop. Translation of ferritin mRNA begins. In contrast, mRNA for transferrin receptor, a protein that is responsible for uptake of iron from outside, has a stem loop at the 3' end. When iron levels are low, the same IRP binds the stem loop and increases the stability of the mRNA. More transferrin receptor protein is made. When iron levels are high, iron binds the IRP and releases the protein from the stem loop. Without IRP, mRNA is degraded and no transferrin receptor protein is made. Second example is to control translation by phosphorylation of EIF2. To assemble the initiation complex, EIF2 first binds GTP and the initiator tRNA and deliver the tRNA methionine to the small ribosomal subunit. After the complex finds the initiation codon, GTP is hydrolyzed to GDP. EIF dissociates from the complex and is recycled. Recycle of EIF2 is catalyzed by a protein called EIF2B. This reaction is blocked by phosphorylation of EIF2. Phosphorylation is catalyzed by kinases that are activated in response to stress, such as amino acid starvation and viral infection. Therefore, phosphorylation of EIF2 inhibits translation at the initiation step. Finally, gene expression can be controlled at the protein activity level. The activity of proteins depends on chemical modifications. Proteolysis and phosphorylation have been covered in lecture B05. Gamma carboxylation occurs on glutamic acid residues, producing calcium binding sites. For example, generation of carboxyglutamate residues on factor 9 and 10. Acetylation is addition of acetyl group to a protein. For example, histone H3 protein can be acetylated on lysine residues. Granulation is addition of farnesia or geranium-gerania lipid groups to cysteine residues of membrane-associated proteins, such as Ras and Rab small GDPases.